Cell membranes play a key role in cellular signaling. This is for two key reasons. First of all, since our cell membrane acts as a barrier for our cells, it controls what molecules move through, including signaling molecules. Secondly, our membrane often acts as kind of a signaling platform for our cells. If we recruit molecules to the membrane, that can then make it easier for those molecules to find each other and interact in order to trigger signaling pathways, compared to if we have those molecules just sitting in our cytosol trying to find each other within that larger 3D space. Because of the importance of the cell membrane for signaling interactions, it's very important that we have lipids that are involved in these interactions as well. So biologically active lipids, or also called signaling lipids, are lipids that are going to be present in small amounts and will play key roles in these signaling interactions that occur around our membranes. So that the advantage of using lipids here is that they'll often be able to pass easily through our cell membrane, so they can be key for passing signals through the membrane. And of course, they can also sit within the membrane and then interact with other molecules that are coming to our membrane as that signaling hub. These biologically active lipids include things like lipid-soluble vitamins, so our vitamins A, D, E, and K, as well as a variety of other important lipids. As we've seen, there's a wide variety of different lipid structures. Our signaling lipids will be no exception to this. So we'll see a very diverse set of different lipid molecules used as biologically active lipids. Let's start with our phospholipids and sphingolipids. So of course, we've already seen these as membrane lipids. So our phospholipids, of course, have two fatty acids that are linked through ester bonds to a glycerol backbone. And then we have a head group that is attached to our third alcohol of our glycerol. And this head group itself will be involved in a variety of different signaling interactions. So, for example, we have signaling lipids such as phosphatidylinositol 4,5-bisphosphate. It has multiple negatively charged phosphate groups, as well as alcohols that can use hydrogen bonding to interact with various protein partners. We also have a variety of different sphingolipids, especially our glycosphingolipids that are involved in interactions as well. In that case, of course, using those sugar groups to interact with key proteins. Many of these membrane signaling lipids will also influence cellular signaling through their breakdown products. So we saw how we have various phospholipases that will cleave these ester bonds within our phospholipids in order to release smaller molecules from our original phospholipid. So this is used both for recycling our lipids as well as creating new signaling lipid molecules. One key example of this is with our phospholipase C that targets our PIP2, or phosphatidylinositol 4,5-bisphosphate. So phospholipase C cuts our ester bond here to completely remove our head group. So in that case, we end up producing diacylglycerol, where our two alcohol groups are still attached to fatty acids, but we have our one free alcohol group where our head group used to be, as well as our inositol trisphosphate, which is just the remaining head group of our original PIP2. Our diacylglycerol here will then diffuse through the membrane and interact with protein kinase C as it's recruited to the membrane, while our inositol triphosphate will trigger calcium release from the endoplasmic reticulum. Both of these events will increase the activity of protein kinase C and lead to signaling cascades within our cells. Phospholipases can also trigger signaling events by cutting off specific fatty acids from our phospholipids. So phospholipase A2 cleaves the second fatty acid, which again is usually our unsaturated fatty acid, off of our phospholipids. In this case, we have arachidonic acid here as our polyunsaturated fatty acid. And arachidonic acid is a very important signaling lipid because of the variety of different biologically active lipids that we can produce from arachidonic acid. So after our reaction, we end up creating a lysophospholipid, which is just a phospholipid with one fatty acid chain cut off. And we also release our arachidonic acid. Arachidonic acid will go on to form several other lipid signaling molecules, which we'll look at in more detail in just a minute, while our lysophospholipid can also go on to make other biologically active lipids. So our lipases can affect lipid signaling in several different ways. First, of course, they're involved in recycling or removal of lipids from our membranes. So if those are carrying out some specific signaling role, our lipase would dampen that particular signal. Secondly, our lipases can directly create molecules that will affect our signaling pathways, such as what we saw with our phospholipase C that produced our diacylglycerol and inositol triphosphate to then trigger that activity of protein kinase C. And then, of course, we have our third example here where we are producing products that are then used to synthesize other signaling lipids. Arachidonic acid is a key precursor for many different signaling lipids. So through various enzymatic reactions, we can take our arachidonic acid or arachidonate and convert it into prostaglandins, which are involved in inflammation and fever. We can convert it into thromboxanes, which are involved in formation of blood clots. We can convert it into leukotrienes, which are involved in smooth muscle contraction. And we can also convert it to lipoxins, which have an anti-inflammatory role. In each case, notice we're doing a variety of different kinds of addition and oxidation reactions. So we're using these key double bonds on our arachidonic acid as our reactive sites to create these various signaling lipids. So whether we're using those to create ring structures and oxidizing those to create various alcohol or carbonyl functional groups, or creating epoxides, or just adding on different alcohols to our existing double bonds. The enzymatic pathways to create these various signaling lipids are often targets for various drug molecules. For example, our non-steroidal anti-inflammatory drugs will target the signaling pathway from arachidonic acid to our prostaglandins and thromboxanes in order to reduce inflammation. Aspirin, of course, is a key example of this. So aspirin inhibits the prostaglandin synthesis by acetylating cyclooxygenase. So cyclooxygenase is involved in oxidizing arachidonic acid. It creates one of those cyclic groups within the prostaglandins and the thromboxanes. Within the active site of our cyclooxygenase, we have a key serine residue. And this serine can react with aspirin to create an ester. So aspirin has this key ester group that can transfer our acetyl group, marked in red, onto our alcohol of our serine. So we end up producing our byproduct of aspirin, salicylate, as well as our inactive enzyme here. This, of course, would be an example of an irreversible inhibition because we've covalently attached this group here in our active site to block our normal interaction with our substrate. And so in order to eliminate this inhibition, we would actually have to produce more of our cyclooxygenase. In addition to the phospholipids and sphingolipids that we've looked at previously, we also have a variety of other rare membrane lipids that are involved in signaling pathways. This includes ether lipids, such as our plasmalogen. So our ether lipid, as you'll notice, is a very strange lipid where instead of our glycerol backbone being linked to fatty acids through ester linkages, we have an ether-linked bond to our carbon chain here. So this is no longer a typical fatty acid because we don't have our carboxylic acid group that's linked to our alcohol. But other than that, it still looks like a normal phospholipid, right? We still have a phospholipid linked at our second position, and we still have our normal phosphate head group. This is our phospho. So our plasmalogen here is commonly found in vertebrate heart tissue. It's also found in some various bacteria and other organisms. The function is not particularly well understood. Now, of course, with this ether-linked lipid, it then becomes resistant to our common lipases, right? So our lipases are usually targeting ester bonds specifically and won't be able to target that ether-linked bond. It does potentially lead to some changes in membrane structure, and it can also act as a potential source for other signaling lipids. But again, it's not known for sure what exactly its full role is. One example of an ether-linked signaling lipid is our platelet-activating factor. So notice we have our ether-linked carbon chain here, and then we have an ester-linked group at our second position, although this is an acetyl ester and not a real long fatty acid. You might recall this platelet-activating factor was one of the byproducts of our lysophospholipids that we looked at as a product of our phospholipase A2. Right, so phospholipase A2 cut off this group here, so that then we can end up attaching our acetyl group rather than our long fatty acid. Platelet activating factor was actually the first signaling lipid to be identified. So as the name suggests, it stimulates aggregation of blood platelets and plays a key role in mediating inflammation. So far, we've been nearly exclusively focused on our complex lipids, right? So these are all of our lipids that contain fatty acids, including things like our storage lipids that we looked at earlier, as well as our phospholipids that we see involved in our signaling interactions. That can be our glycerophospholipids or sphingolipids, as well, of course, those strange ether-linked lipids as well. Our second main class of lipids, our lipids that don't contain fatty acids, will also play a key role in many different signaling interactions. So next, we'll take a closer look at these more diverse structures of our lipids and how they're involved in signaling. As I mentioned previously, our biologically active lipids will include our fat-soluble vitamins. So I want to take a look at each of these different vitamins so we can see what their role is in our bodies. So vitamin D is actually synthesized from cholesterol, which we'll be looking at more closely in our next lecture. And it helps to regulate our calcium metabolism. So I'm sure you've heard, of course, how vitamin D is critical for building bones, and that's through this calcium metabolism-based mechanism. So our vitamin D metabolism starts with 7-dehydrocholesterol, our structure shown over here. And UV light is actually required for this metabolic pathway. So that's one of the reasons why getting sunlight is so important for us. So the UV light in sunlight will help to generate vitamin D by actually breaking this shaded bond right here in our 7-dehydrocholesterol. So this, of course, occurs in our skin where we have that UV light exposure. This creates vitamin D3, which is then further converted into calcitriol, which acts as a key hormone to help control calcium metabolism in our kidney, liver, and bones. So, of course, that's why we talk about vitamin D as being important for strengthening your bones, because calcium is so critical for our bone structure. Our next fat-soluble vitamin is vitamin A. Vitamin A is derived from beta-carotene. So as you can see, our structure here is quite different from the structure that we saw for vitamin D. Vitamin A is involved in vision and it also acts as a precursor for various other hormones. So in order to form our vitamin A, we break down this beta-carotene and we form this all-trans retinol, or vitamin A1. This can then be oxidized to our retinal. So notice how our ending name there goes from a retinol, indicating our alcohol, to our retinal, indicating that aldehyde. And we create this single cis double bond which can actually be converted to a trans double bond by visible light. So absorb visible light and convert this to all trans retinol, and that is then used as a signal to indicate that we have absorbed light and we should be seeing something. On the top, of course, you can see how our all-trans retinol can also be converted to all-trans retinoic acid and can then be converted into that hormonal signal that I mentioned previously. Vitamin A, as well as many of our other biologically active lipids, are classified as isoprenoids. So isoprenoids are synthesized by condensation of multiple isoprene units. So you can see the structure of our isoprene over here. So it's a conjugated diene, where we have those two double bonds next to each other, where they would conjugate to each other, as well as our fifth carbon as a branch up here. So we'll combine these 5 carbon units together in order to form various lipids. Even cholesterol itself is actually formed from individual isoprene units. Plants will actually often tend to use smaller isoprenoid molecules because this will lead to a low boiling point and relatively volatile compounds. So our actual isoprene right here, of course, with only 5 carbons, will tend to have relatively weak intermolecular interactions and that low boiling point then. Our larger isoprenoids, of course, will not have low boiling points because our boiling point, of course, will increase as we increase our molecular weight. These small isoprenoid molecules can then be used to communicate between plants through the air. So they'll be able to evaporate from one plant and then travel through the air to reach the other plant, or of course, other organisms as well. Our other lipid-soluble vitamins, vitamins E and K, as well as some of our other signaling lipids, will all use this basic isoprenoid structure. So you can notice in our lipid structures shown here, we have a repeating 5-carbon isoprene unit. So our little dotted lines here show the divisions to mark our different isoprene units. Note the double bonds have been reduced here, so we just have our alkane carbon chain. And this will vary between our different lipid structures. So we do have a double bond left here still in vitamin K, but of course not in every single isoprene unit within vitamin K. The isoprenoid structure of these vitamins mostly serves to create a very nonpolar anchor for each of these vitamins. The key chemistry of our vitamins occurs in this cyclic head group for our vitamin E and vitamin K. Now, of course, the head group itself is still relatively nonpolar, so this isn't a true polar head group like we see with phospholipids. But this is where most of the interesting chemistry will occur for the actual behavior of our vitamins. So you'll notice that warfarin, which is a blood anticoagulant, actually targets that structure there. So you see it has a similar looking ring structure to try to bind to vitamin K enzymes and prevent the recycling of vitamin K and in order to prevent blood clotting. So as long as we match kind of that key head group structure, we don't have to exactly match our isoprene chain here for our drug molecule. So vitamin E will specifically counteract lipid oxidation. So especially when we have unsaturated lipids in our membranes, these can react with reactive oxygen species, such as our hydroxy radical shown here, to create a variety of lipid radicals. So you might remember from organic chemistry that sites adjacent to double bonds are extra reactive because of the conjugation that they have with that double bond. So we can create a radical, which can then react with oxygen to create a peroxy radical. This can then propagate and create additional radicals, as well as creating our lipid peroxides. So this sort of oxidation product will slowly turn our unsaturated lipids into a variety of different peroxide-based products. Now, this, of course, will affect the properties of our unsaturated lipid, and can ultimately then affect the properties of our membrane as a whole, as more of our unsaturated lipids are turned into these lipid peroxide products. Vitamin E will act to counteract this by attacking these radicals and trying to eliminate these radicals that would otherwise react with our unsaturated lipids. So we use this key phenol group on our vitamin E to react with those peroxides and create a radical on our vitamin E. This can then be recycled through our usual antioxidant pathways. So that's where we have glutathione involved in order to eliminate that radical and keep our body safe from these sorts of reactions. Another example of our biologically active isoprenoids is our lipid quinones. Ubiquinone and plastoquinone are both involved in electron transport. So this is a key part of our metabolism, where we need to take electrons and use those to reduce oxygen to make water, and the energy from this process is used to help fuel our body. So again, notice we have our key isoprene units here, in this case with double bond for each of our units, and then we have our head group that actually undergoes the chemistry for our biologically active lipid. We also have dolicol down here, which is a sugar carrier. So we have our free hydroxy group that can attach to sugars through our usual glycosidic bonds, and then it's used to help attach sugars to our glycoproteins and glycolipids. Again, the alcohol here is our key reactive group, and we have our isoprenes to create that nonpolar lipid chain. The final class of biologically active lipids that I want to look at is our polyketides. So polyketides are a very diverse family of compounds that are formed from alternating ketones and methylene, that's CH2 groups, and then we reduce those in order to create a variety of different products. So notice in most of these products you'll have alternating, so here we have alcohol, so this would be our ketones reduced to those hydroxy groups. We also have double bonds, which of course can be from reduced ketones as well, all in kind of this alternating pattern. Now, as you can tell, the structures of these polyketides can be very diverse, right? So we have three different structures here with three different functions: an antibiotic, an antifungal, as well as a statin here that helps to target cholesterol metabolism. And each of these look very different in structure. Again, the main thing that you see is you see this kind of repetitive pattern of functional groups sticking out, often either hydroxy groups or ketones or even our alkenes. The diverse pattern of what functional groups we have involved from our original polyketide structure then, of course, leads to our various specialized functions. Now, we won't look at any of these in detail. I just wanted to show you these as an example of some of the different diverse lipid structures that we have involved in our lipid signaling. So again, with our biologically active lipids, we really see the full diversity of our lipid biomolecules. We have things ranging from our more typical complex lipids with those fatty acids linked to glycerol or sphingosine, all the way to compounds like our polyketides, where we just have a wide variety of very different looking molecules just based off of kind of this repeating ketone methylene pattern. Now, you might have noticed that there's one key lipid that we've left out of our discussion of biologically active lipids, and that is cholesterol. So cholesterol, of course, is a very important and very well-known lipid. So, of course, it gets a lot of negative press because of its role in heart disease, but it also has very important, very critical functions within our body. So we're going to look at sterols and steroids in our next lecture to take a look at all the different roles that we see there with that class of lipids.